Hello and welcome to this lesson for Advanced Higher Biology, which today is looking at the process of meiosis. So, what we're looking for you to understand is how the process of meiosis can produce genetically varied gametes for the organism. And you need to be able to define the process, describe what happens during the first stage of the process, explain what we mean by crossing over, random assortment, and how these increase the genetic variability of the organism, and you need to be able to describe what happens in the second stage of the process. So to begin thinking about what meiosis actually is, essentially it's dividing the nucleus and producing haploid gametes from a diploid gametocyte, and a gametocyte would just be essentially the gamete mother cell, as it may also be called. It's the type of cell that's going to be able to produce either eggs or sperm, or pollen or ova if we're looking at plant cells. So this one here would be considered our gametocyte. It needs to divide. We go through two divisions, and we're producing these haploid gametes, or eggs or sperm, or pollen or ova, depending on what type of organism it is that's going through the process. So to quickly go through some general terminology that you should be aware of already. Thinking of humans, the human body has got 46 chromosomes in the nucleus of every cell, and these are present in 2 copies of each of the 23 types of chromosomes. So from 1 to 23, we've got one copy from one parent and another copy from the other parent. Now, the 2 chromosomes in a pair are referred to as homologous chromosomes, and they can be determined because they have the same size, the same centromere position between the 2 of them, and they also have genes for the same characteristics present at the same loci or locations that they've got available. So we've got genes for, say, eye colour, both present on this chromosome, say this chromosome number 1, and the genes are present on the same place on each homologous chromosome. And the homologous pairs are found because one copy of each chromosome has come from the female parent and the other copy has come from the male parent. So because they're inherited from different parents, the alleles of those genes on each homologous chromosome might be slightly different. So a single set of chromosomes will come from the female parent in her gametes. Another single set of chromosomes will come from the male parent in his gametes, and as these fuse together to form a diploid zygote. So each gamete cell has one single set of chromosomes, and that's referred to as being haploid. So here we've got 3 non-homologous chromosomes, and then when that has fused with the other gametes, we have our diploid zygote, where it's got a cell with a full double set of homologous chromosomes and is therefore referred to as diploid. So in most organisms, the majority of the life cycle is spent as a diploid multicellular organism. Meiosis is what's going to help produce our genetically variable haploid cells, and these develop into gametes, fuse with another gamete from another individual, are fertilised. These haploid nuclei produce a diploid zygote. We then have mitosis taking place to produce genetically identical diploid cells, and that produces our multicellular organism. Now, meiosis is essentially a way of reducing the number of chromosomes in each cell. It's not really a cycle in the same way that mitosis is. It has 2 individual divisions, so we're starting off with a duplication of chromosomes. We have one division to allow the homologous chromosomes to separate, so the parent pairs to separate, and we have a second division to allow the sister chromatids to separate, and we're going to look at both of those. So the first stage of meiosis begins with the duplication of the chromosomes to form 2 genetically identical chromatids which are attached at the centromere. So if this is our parent cell, our original diploid cell, this individual has got 2 sets of homologous chromosomes, chromosome 1 and chromosome 2. So this is our homologous chromosome set, one of these chromosomes from the parent of this individual and one of the chromosomes from the other parent of this individual. And these have duplicated, so we now have 2 chromatids held together at our centromere, and that allows us to then start dividing our cell to produce our haploid gametes. We then have the chromosomes condensing, and our homologous chromosomes are going to pair up. Something called chiasmata form at points of contact between the non-sister chromatids of a homologous pair. So thinking about what that means. If looking at this chromosome, for example, this is one sister chromatid, this is the other sister chromatid, and the purple one over here is the homologous pair to this chromosome. So we have these chiasmata forming, that's points of contact between the non-sister chromatids of the homologous pair. And what happens there is we wind up exchanging sections of DNA. And linked genes can maybe present on these chromosomes, and this crossing over can result in new combinations of the alleles of these genes. So have a little look at crossing over in a little bit more detail. When we have our homologous chromosomes starting to align, starting to pair up within that cell, we may have this chromosome crossover, these chiasmata formed, and as a result we wind up with these recombinant chromosomes. They've swapped some pieces of genetic material. And in terms of gene linkage, what we're really thinking about is the fact that, remember we said that there's specific genes present on different loci of different chromosomes. Well, it might be that we see a similar pattern of inheritance between one allele that's up here and another allele that's down here, and they'd be referred to as being linked. And you might see them because they're on the same chromosome. An individual's quite likely to inherit one's chromatid from one parent, for example. So this crossing over allows us a little bit of variability and gives new combinations of these genes. Essentially, crossing over is very random, and it helps us produce genetically different recombinant chromosomes. So it allows us to produce a little bit more variety in our organisms. We then have our spindle fibres are going to come over, and they're going to attach to the homologous pairs and line up at the equator of the spindle. And this is another version, or another method, of how we wind up with genetic variability within our organisms. This is referred to as independent assortment. So essentially, when the chromosomes are lining up, and the homologous chromosome pairs are going to line up across our equator, the order in which they do that can be varied. So, for example, if looking at this one, all of the ones that were the paternal chromosome are lining up down here, and all of the ones that were the maternal chromosome are lining up down here. Now, it's important to remember, while we're talking about this, that when we're referring to maternal and paternal chromosomes, that's the chromosomes inherited by this individual from their parents. So this could be one individual who is a female producing egg cells, so they will all obviously all become maternal chromosomes when she passes them on. But when we're looking at the assortment, when we're looking at the arrangements that she's passing on to her offspring or producing into her gametes, we're looking at her inheritance. So this is the ones she inherited from her father, and these would be the ones she inherited from her mother. Now, when we look at independent assortment, what we can see is, for example, one way of arranging these ones is all the ones from one parent down one side and all the ones from another parent down the other side. However, there's no reason they would necessarily line up like that. All the homologous chromosomes are lining up with their pairs. They're not lining up in comparison to the ones above or below them. So you could also have this arrangement where we've got two from the paternal chromosomes on this side and two from the maternal chromosomes on this side, but it's the top and the bottom one. Here we've got the top and the middle one from the maternal one on this side, and here we've got the middle and the bottom one. So there's a good number of different ways in which we can arrange these particular chromosomes, and this is referred to as independent assortment, where essentially each pair of homologous chromosomes is going to be positioned independently of the other pairs, irrespective of their maternal or paternal origin. Once we have these chromosomes lined up, we're then able to look at separating them. So similar to in mitosis, the chromosomes of each homologous pair are separated and are going to move towards opposite poles. We then have the nuclear membrane forming around them, followed by cytokinesis occurring and our two daughter cells formed. However, this is only the first stage of the division of meiosis. Remember, our goal at this point is to produce our haploid gametes, and we still have two divided diploid cells here. So what we've wound up with is a little bit of variation. We've got some crossing over has occurred in both of the chromosomes. We may have had some independent assortment. So here we've got one from mainly the paternal origin, we've got another one from mainly the maternal origin, and the opposite on the other in the other cell. So we still need to go through our second stage of division, meiosis II. And essentially all that's going to happen in this one is we have a similar idea. There is no crossing over this time, as we don't have the two homologous chromosomes in the same cell, and the independent assortment can't really take place either because we've only got one of each set of chromosomes. Remember, this is a sister chromatid and they're attached together by the centromere, so they've got no choice where they're going to line up with or where they're going to pair. So they're going to line up in the middle of the cell again, and the spindle fibres are going to pull those sister chromatids apart into opposite poles of the cell. Our nuclear membrane will form, cytokinesis occurs, and we wind up with our total of 4 haploid cells being produced. And these would be the gametes that would then go on to fertilisation. So if we look at the overall process of how that might occur, remember, this is our parent cell that we started off with, which had 2 pairs of homologous chromosomes present in it. It's diploid. We've then had one division being undertaken and then a further division to produce our haploid gametes as a result. And if you look at those ones, you can see that while 2 of them are genetically quite similar to the original, we've had some independent assortment. So we've got a paternal and a maternal origin, and a maternal and a paternal origin chromosome here, and we've had a little bit of crossing over happening in these ones. So each one of these individuals is going to be it's different from the other and ever so slightly different from its parental cell. So as a result, we've managed to produce from our diploid cells 4 haploid gametes, which can then go on to fertilise another haploid gamete cell produced by another parent, providing a good deal of variety within the organism. So from that, you should be able to define the process of meiosis as being the production of our haploid gametes from an original diploid gametocyte. That during meiosis I we have duplication and division of our chromosomes to separate them out. We also have crossing over and random assortment occurring in meiosis I, and that crossing over is where we swap sections of DNA from one homologous chromosome to another homologous chromosome. And that random assortment is where it considers the positioning of the chromosomes as they line up across the equator, and the fact that they line up independently of each other, irrespective of their maternal and paternal origin. And that these increase variability because it allows the haploid gametes to look genetically dissimilar to the original gametocyte. That during meiosis II, we have a further division during which the sister chromatids of each chromosome are separated to produce a total of 4 haploid cells.